Welcome to our coverage from Bioprocessing International European Summit here in Vienna. At our co-located cell line development and engineering conference, it's an ideal opportunity to place a finger on the pulse of the cell line industry and to find out the latest happenings and latest technological advances being made. We spoke to Daniel Galbraith and to Jonas Schoenduber, CEO of Cytina. Here's what they had to say.
I think here at the conference, some of the new cell line development technologies and really trying to speed up taking cell line development to market has been sort of like something that I've really been surprised at how much effort's going into at the moment. So there's lots of different presentations sort of showing how we can reduce the time down from maybe 12 months to 6 months and get a product into the clinic much faster.
What I've learned here a lot, especially in the first day of the cell line conference, was really that the FDA is looking for a proven clonality for any cell line that can be used commercially or in a therapeutic environment. And I think that's a biggest concern of the regulatory bodies and therefore also a lot of the biopharmaceutical companies. So I think, one of these challenges is to prove that the cell line that you're working with, that you scaled up and now are ready for production, is clonal and has always been clonal. So we offer a technology, a benchtop-sized instrument that allows you to isolate individual cells and also give you an image-based proof of that clonality. So right from the start, you're working with a proven clonal cell line. So it's a benchtop-sized instrument that works fairly similar to an inkjet printer. So we dispense minute amounts of liquid in a droplet. And other than an inkjet printer, we supply an empty cartridge and the customer fills it with this cell suspension. It's ink, right? And as opposed to a regular inkjet printer, we have a microscopic-based imaging system that allows us to image the nozzle and see if there's an individual cell and only dispense those droplets that will contain a single cell into the substrate, such as the well of a micro well plate, 96 well plate, 384 well plate, such as that. So we have several parameters that we derive from the images, such as size, roundness, contrast, and we typically work with a high roundness if we want to go for a high quality cell. And then we measure the viability, the clonal recovery rate in the place afterwards. And that's also what our customers do. And they typically, or I guess all of our customers see a significantly increased viability and clonal recovery rate when using our technology, as it's designed to be very gentle on the cells. Very important for us and for our workflow is to have a high quality sample with a high viability to start with, and then we designed the entire chip design, instrument design to be very gentle on the cells. So there's no external pressure, there's no lasers, there's no electrostatic charge, so there's no external stress that might deduct the viability of the sample. And if you start with a good sample, you'll end up with a very high viability later on in your plate. So what we typically call the single cell printing efficiency is how many wells of a well plate will actually contain a single cell, and that's typically way above 90%. And even for those where there might be a void droplet without a cell, you still have our nozzle images placed on the computer, saved on the hard drive in a right protected format. And this allows you to identify these wells and also prove that there has been no cell dispensed into these wells.
Want to know more? Well, head over to our blog or get in touch via social and join in the conversation. Make sure to check out the website in the link below to watch all the highlights and more interviews from this year's conference. And we look forward to seeing you next year in Amsterdam.